Hi everyone. Let me welcome you to my poster presentation titled Genetic Determinants of Apparent or Possible Adrenocorticotropic Hormone Resistance in Asthmatic Children on Corticosteroid Use. In the management of asthma, inhaled corticosteroids are very potent medications that are used, but these can lead to hypothalamic pituitary adrenal axis suppression. Adrenocorticotropic hormone resistance is the body's inability to respond to ACTH stimulation. Now, both HEPAS and ACTH resistance may have similar clinical symptoms. So it is important to determine genetic markers that are specific to ACTH resistance as against HEPAS, which is vital for personalized asthma management. So the aim of this study is to identify genetic variations that may predict possible ACTH resistance or protection against it. A cross-sectional study was carried out involving 96 asthmatic patients on inhaled corticosteroids. This flow chart shows the methodology that was employed in this study. After administering miterapone, clinical samples were collected, including saliva, where the DNA was extracted and sequenced by ion torrent sequencing. Association analysis for single nucleotide polymorphisms were conducted and controlled for age, sex, the drug dose, and BMI z-scores. Significant SNPs were annotated, filtered, and then further prioritized. This table shows the results for the association analysis. The basal ACTH cortisol ratio as well as post-miterapone ACTH cortisol ratios were used as measures for ACTH resistance. The top two SNPs were significantly associated with resistance with the positive beta values. The next two were associated with protection as indicated by the negative beta values, which is an inverse relationship. SNP RS 14235 lost significance after correcting for multiple testing. The last two SNPs are inconclusive because the beta values are conflicting each other. So the heat map here just shows you the expression levels of the significant genes across various tissues. Strikingly, C2ORF53 had a very low or practically no expression level in all the tissues compared, except in the testes. But we know that genes can be switched on during certain environmental conditions, such as the use of inhaled corticosteroids. And so this SNP and the genes need further investigation. These box plots represent a genotypic comparison of the mean values of ACTH cortisol ratios. The green boxes represent homozygote variant genotypes. The blue represents the heterozygous, while orange represents homozygous variant genotypes. The red horizontal lines represent the confidence interval of the mean values, with the mean values displayed as dots in the box plots, while these numbers represent the actual mean values. So if these horizontal lines do not overlap, it means that adjacent boxes are significantly different from each other. So, for example, RS6962 means that carriers of the AA genotype have a high basal ACTH cortisol ratio compared to individuals who are GG or GA carriers. Succinate dehydrogenase A subunit encodes a protein that is integral to mitochondrial respiration, and mitochondria are known to produce cortisol in the adrenals. The impact of zinc finger protein 668 on the HPA axis is not known, but we know that other zinc finger proteins like that of 764 is a cofactor for glucocorticoid receptor. Similarly, gene PRR330 encodes a member in the family of proline-rich proteins, and like other proline-rich proteins, it may regulate steroid receptor function. And so, mutational distractions of these genes could have an impact on glucocorticoid binding at the adrenal or selectively at the hypothalamic pituitary level. To conclude, in this study, 2 SNPs were linked to ACTH resistance and 1 being protective against resistance. SNP genotype validation and functional studies are recommended to confirm the functional role of these SNPs in the axis. I would like to acknowledge the support of my supervisors and others who contributed to this work. I'm especially grateful to my funders and ISCB for the travel fellowship. Thank you.